home, online and offline. It is an honor to welcome a distinguished scientist and accomplished academician as our session chair, Professor Dr. Rupesh Deshmukh, a professor at Central University of Haryana. Before joining the university, he was Ramalingam Swami Fellow and Associate Professor at Plaksha University, Mohali, India. He has obtained his PhD degree in Agriculture Biotechnology from SRTMU, London, India. Dr. Deshmukh did his first postdoctorate at University of Laval, Quebec, Canada, where he worked on the molecular mechanisms involved in silicon uptake in plants. He identified silicon transporting aquaporins in several plant species and efficient exploration of omics resources. He did another postdoc at Missouri University, USA, where he was involved in researching focusing on translational genomics and the development of genomic resources for crop plants. Subsequently, he joined back at university level as visiting professor. At present, his group is working on different challenging genomes editing projects. He received several highly competitive awards, including Ramalingam Swami Fellowship Award, Dr. P.S. Khangoje Gold Medal, and NASI Scopus Young Scientist Award. He is recognized as a fellow of ISGPB and ARRW. Recently, his name is featured in the list of world 2% of scientists published by Stanford University for the years 2021, 2022, and 2023. He has published over 100 papers in peer-reviewed journals, over 1,560 citations, and a remarkable H index of 55. He is an editorial board member for several highly reputed journals like Journal of Advanced Research, The Plant Genomes, One, BMC Biotechnology, and Frontiers in Genome Editing. Thank you, sir, for joining us for 8 AMIFOST 2025. Can you give, please? Two words and then we'll start with the section. And then I'll read out the brief of everyone.
Okay. Good morning, all. I'm really thankful to organizing committee for giving me this opportunity to chair this interesting session. The speaker line is very impressive. Mr. Aman Chaudhary, Dr. Rachna Sherawat, and I hope you will also enjoy these experts. And I really wanted to thank Dr. Durgesh, who really always there for me, and he gave me such opportunity to visit a meeting. Dr. Renu Khedkar, Dr. V.K. Modi, Dr. Shimurthy, Dr. Bhatnagar, and Dr. Monica. I'm really thankful for this, and I'm always interested to visit MIT because of the environment here, which is very open. That is open science environment. A lot of opportunities there to interact with different peoples. Infrastructure wise, you already have seen this campus is very beautiful, and MIT have very good expertise in all different frontline domains. So I hope you will interact with the different faculties, different students, and get whatever out of you can achieve from this conference. So thank you very much.
I extend my warm welcome to our speaker, Mr. A.K. Bhatnagar, Technical Head, Anmol Industries, to kindly speak on current trends in food science and technology. Especially the future of India. You people who deserve to be applauded for being developed by MIT University. Congratulations to the management and the staff of MIT University.
Actually, the session was supposed to be delivered by our director, Mr. AMIFOST, but somehow he couldn't come. I am taking this opportunity to talk to you. It is my privilege because I've got more than 55 years experience in biscuit industry. Actually, I retired from Britannia industry after working 36 and a half years. Then I went abroad and in today for last 17 years I'm assisting on industry at Greater Noida. I always loved to share the knowledge, the experience, what we got it, I got it, to share to these youngsters. I'm very happy because normally we take so many technical sessions, queries, process development, problems, solving problems. It gives a motivation to be in job today. When you share, you get more knowledge and your knowledge is cleared. So I'm just talking about the history about biscuits. Biscuit trend in food science and technology are evolving to meet consumer demand. If you see, in 1980 to 2000, wheat biscuit was very popular. Then, because market wanted that, liked it, there was no innovation. But if you see today, glucose biscuit has got some real nutrition ingredients. Till today, the newly born child, the first food is glucose biscuit dissolved in water, all mothers used to learn. So it is still very, very relevant and worthy. From 2000 to 2010, there is a change. The people wanted some change. Consumer wanted some change. The investors wanted change. Then it butter biscuit came in the market, which was prominently dominated the glucose biscuit. From 2010 to 2020, the cracker biscuits began to the market because the consumer like it because they find it is more crispy and tasty. So the change is having place or the trend is in the market changing due to the consumer's demand. Now from 2020 to 2025, there is a trend change in the industry. Today the people want more protein biscuits because new generation has got a different eating habits. They're liking biscuit is being not like people like going to gym etc. where their coaches says have protein powder. But the problem is that protein, if you powder it, is very costly. The cost is one of the factor. But for the market like India and some other countries, in export also, the people are asking for a protein-rich biscuit. And today, the biscuit industry is more focused on protein-rich biscuits. This is the present change is coming, and the market is also demanding. The major market demand is going to be the foreign countries like Africa, etc. They are wanting more protein biscuits. So the point what I just want to, there is always a change in the food industry, and with that, the changes are happening. If you see in 2080, we were in Britannia were having a plant and equipment imported from the Britain, and we started very simple way of making the bread by putting yeast and normal nothing. But gradually it was changing. New improvements have come. New things have been evolved and the product because the old, but the basics have remained the same. If you see, in good old days, the flavor, aroma of the product are different. Today, when we go for more commercial, fast moving product, the things are changing. Now the high speed mixers have introduced. Earlier used to be the slow speed mixers. With high-speed mixers, we are saving time. We are saving fermentation time. We are developing the products more effectively than the old system. So there's a change, innovation is going on. Up today, if you are seeing today, I find a lot of change. Anmol has got actually Anmol started in 1994 from Calcutta. And today we have got 10 factories working around India. After that, two are at one at Greater Noida and other at Ghaziabad. What I want to share with you, Anmol has put up a very latest technology in biscuit. The plant they have put in Thakurganj, totally automatic. Where we used to see there's a change. Why change? Because today the manpower cost is going up, so the automation is required. Lot of things have changed, lot of improvement. I mean, the food science also changed. Lot of good advice has come to develop the pure gluten, good product, good texture, good baking. So good ovens have been there. But what is more important is basics remain the same. Basics means process. If we maintain the same, we cannot go apart from the basics of the things. Anything you want, if you have to be with some basic within your knowledge. With today, earlier we used to weigh the dough. Suppose you have to do a weight of 400 grams today, you need not to weigh. You need not to put a people. Now today the mechanism is there. If the weight is less, it will be automatically kept aside by machine itself. So the improvement is coming. So the innovation has come. So innovation coming where we were unique doing the force pulling. When the force pulling was just putting the products in front of the cooler or something. Now today is automatic. So the different zones have been developed with how to cool the bread or how to cool the biscuit. So the things what we were doing earlier today has changed. But basic focus came from 2020 to today. The major change in the industry has come at the hygiene. The process, if you see the all factories coming up, have got a very high standard of hygiene and cleanliness. Today, we feel people are making the factory just like a five-star hotel. Because anybody who's coming for the conversion of their product or to ask them to make the product, they see the first hygiene. So health consciousness has become a very, very important nowadays. Hygiene is the top priority in the food industry today. Anybody earlier used to be people just manage it how to make it the rusk or bread or biscuit. But today, more precautions, government regulations, following the people. So the system has improved. And that way, what we see, Amity has done a great job in developing the young people for understanding the food industry and product. Some of your students have come to Anmol. They have done internship and they have done a very good job. And we have absorbed them. So on this platform today, I'm welcoming you all. If anybody wants internship in Anmol, you are welcome. If you do well, we will accommodate you. We will provide you the job. This is my commitment to you because since I am in the industry, I'm not only Anmol, but lot of factory people knows me, Bitaya people knows me, so we can help you out to get you a internship or job if you want. We are ready to give you and provide you. With all this good wishes, thank you so much.
Thank you, sir. Our next speaker has joined online, Dr. Rachna Shehrawat, Assistant Professor, Department of Food Process Engineering, NIT Rourkela. Ma'am has published over 50 research articles in national and international journal of repute, 22 book chapters, and authored 3 books. With that, I extend my warm welcome to Dr. Rachna Shehrawat. She has joined online, sir. आ नहीं रहा अच्छा नहीं है इस सेशन में हाँ वो करेंगे ना नहीं सर हाँ नहीं करना मीटिंग में ये देख लिया मैंने ज्वाइन हो नहीं मैम नहीं मैम कॉल कर रहा हूँ उससे हाँ रचना ज्वाइन नहीं किया अभी आपका अनाउंस हो चुका है प्रेजेंटेशन अच्छा 5 मिनट्स में बता दिया मैंने अभी क्या करना है So there is some technical glitch. Our online speaker will be joining in another 2-3 minutes. Yes, dear participants, there is a small, I think, technical issue, and Dr. Rachna will join soon now, just to give it 2 to 3 minutes. But in meantime, just I want to share few things with all our participants. Our conference partner Anmol just now addressed you, and for students of the food technology, it's a great news that they will offer job opportunities as well as the internship. So that is a very good sign of the industry academia partnership. So we hope that with this, they wish that all the our students, participants, and those who are looking for internship, please contact our coordinator, Dr. Ashish, and we will coordinate with the Admol industry. And it is a very good brand for the biscuit. Just want to
have you thought any time that there are the two synonymous word, biscuit and cookies? What's the difference? Have you just given a thought that join kar liya usne? You just give a thought on this particular these two words, and third word is the uh, crackers. Crackers, okay, it's a little different word and different side, but it is a again category of biscuit only. But difference between cookies and biscuit, you must have read in your courses, or as a common man, yes, we are jumping on cookies because that word sounds a little more attractive to our tongue. Otherwise, it is a biscuit only. I didn't find much difference, though scientifically or other way it is given some difference that biscuits are flaky in texture and more crispy than the cookies. That is, so we can say that there is a little difference in the processing technique, ingredient technique, but literally I say both are the same. So generally that I think one is American concept and one is British concept. So anyway, that is a part of your now innovation for with this conference, that how you discover these two words in your dictionary. Just try to give a thought and then come out with this. What are the difference? You won't find much difference in literature as per I know, but anyway, so this is a part of a debate for this, our all the students or the consumers. As he has quoted the example of biscuit, those days only one biscuit variety or two biscuit variety, five biscuit variety, and there are now infinite number of the varieties. From where it has come? All it has come from your food science. All it has come from food technology, from science background, and that we keep on developing new products. That is our tendency of the food technologist to give a new concept, new flavor, new texture, new color, new packaging, and new ingredients. Everything it is coming. Why it is coming, that we know everything, that because that is a business opportunity in this sector. I think that is the most important to look with this aspect, that yes, this consumer wants change in the food habit. It is essential. Industry wants to market, keep themselves alive with the consumer. That's why these changes we are looking around us. Okay. So now Dr. Rachna has already joined. So I don't want to come in between now. So Dr. Rachna, please.
Yes,
Can
sir.
you hear me?
Yes, sir. I can hear you. Can you hear me, sir?
Yes, please. Thank you.
A very good morning, sir.
So, as our online speaker has joined, Dr. Rachna Shehrawat, Assistant Professor, Department of Food Process Engineering, NIT Rourkela, India. Ma'am has published over 50 research articles in national and international journal of repute with 22 book chapters and authored 3 books. With that, I extend my warm welcome to Dr. Rachna Shehrawat. Thank you, ma'am.
Thank you so much and for giving me a brief about me, and a very good morning to everyone, and hope can you see my screen?
Yes, ma'am.
Okay, thank you. Please give a heads up once the time is over. Okay, so though I'll keep a track of this one, and probably it's very nice to see that you are ahead 5 minutes before as per the schedule. Generally it is late, but in this session it's little 5 minutes early. So very happy to see that. Okay, so today's talk is on technological intervention to reduce anti-nutritional compounds in pulses. Though it's a very common topic, and we have been doing it for the reduction of these one from a long time. But here my major emphasis would be on the novel technologies that have come existence over the time, and how effective they are in reducing the anti-nutritional factor, simultaneously enhancing the nutritional value. Because only reduction in certain factor is not important, it is also important to maintain the quality. So basically, I'm working in NIT Rourkela, and it's in the Odisha. And these are the different views of my campus, and it's a beautiful green lush campus. And I invite all of you to come over here whenever the time permits. will be very happy to host you. And so we have over here different in the Department of Food Process Engineering. We started only in 2013, and it's been hardly 10 to 12 years that we have come across. So the NIRF ranking of the institution is 15 in the engineering, and the faculty and the students are doing a lot of hard work to maintain the different factors into the research area. So we have BTech, MTech, and PhD program over here. And we have different faculty working in the different groups, like on quality and safety, packaging, process design, horticulture processing, or grain processing, and in the dairy sector. And so coming over to the topic, legume pulses and anti-nutritional factors. We know the legumes, we are the largest producers, but still we have to import the pulses also. And in the 2016, the FAO celebrated the International Year of Pulses. A lot of the innovation and the work has been started during that time, and a lot of emphasis is not on produced or is being kept by the manufacturer as well as by researchers to valorize these pulses apart from consuming in our daily habits. Nowadays, we can see lots of the plant-based products are into the market as people are giving more attention to the plant-based product over that of the animal-based product because of the several XYZ reasons, as we all know. And these pulses, because they are rich source of the protein apart from the fiber and the minerals present into these. So these pulses have not only these benefits, apart from that, it has the secondary benefits also, like they can be associated or have been reported to reduce risk of several type of chronic disease and in this one. And they have, the issue is anti-nutritional compounds, but if they are present in lower amount, that's fine. They even have their beneficial aspect. It's not that they don't have any beneficial, but if present into higher amount can create issue. So what exactly is this? Like for an example, we have primary metabolites where we require these, like your these protein, carbohydrates, fats for our basic energy. Then we have secondary metabolites. Okay, those are not directly involved into the growth, but somehow have the important functioning. Like for the plants, these secondary metabolites act as a defense mechanism. They want to prevent themselves against certain pests or from certain animals. So they create these defense mechanisms. And these compounds are known as anti-nutritional. They are able to digest those plants and all, but we do not have these enzymes to digest these factors. That's why they sometimes become toxic to us if present into the higher amount. So the things as a researcher, for us, the role is to reduce these anti-nutritional factors, because not only these we are not able to digest them, apart from that they bind some important minerals, they bind some important compounds that they do not let our body to available to us. That's why it is important to reduce. So that's why, but they have ambivalent nutritional properties. Means if they are present into lower amount, they does not have any bad effect. But if they are present into higher amount, they can have a variety of the effects on our body. For an example, there are like phytic acids, then amylase inhibitors, flavonoids, glycosinolates, protease inhibitors, saponase. These are the different anti-nutritional factors that are present over here. Like phytic acid, majorly it's bind the phosphorus and several other minerals that are there, making it non-available to our body. So we will wonder sometime, I am taking a complete diet, it is rich in the fruits, it is rich into the minerals and the vitamins, but I'm not able to, or still I have deficiencies of these. It is because sometimes these factors become excess in our body, they bind the important nutrition, making them not available to our body. So that's one of the reasons. Like polyphenolic content, if they are present into any product, so they make the product more bitter, astringent into nature, and have lesser stability into these ones. And these are able to bind some of the protein and the amino acids, making them not available to our body. Similarly, like tannins are there. So these tannins are generally present into outer cotyledon, though can be sometimes easily removed by the soaking process, but they can be sometimes hydrolyzable or condensed tannin. So we have to see like which kind of the anti-nutritional factors are present and in which final product or the application I require. Based upon that, we have saponin. These are also anti-nutritional factor, though they have can act as an emulsifier also. Sometimes they are being even isolated from certain compounds in order to have its further application as an emulsifier. But if present into a larger amount in our daily diets, can have a poisoning effect to our body. So that's why it is important that we need to know that once we are providing any product to the consumer, it is free from these anti-nutritional factors. Another category is which bind proteins and these peptide inhibitors or amino acid and do not let them available to our body. And the proteins are very, very important. One of the category is the trypsin inhibitor. These bind the enzymes actually which are important to break down these amino acids into our body. They bind these one and do not let to function them by the different ways and have their different based on molecular compounds. So these are certain inhibitors. So as a food technologist, as a researcher, our strategy is to reduce these anti-nutritional factors to a amount where it should not cause any bad effect to our body and let the nutrient retain in our body. So one of the very simplest processes are like milling, fermentation, germination, autoclaving. These are the overall different strategies to reduce these factors. Some of the here I have taken an example of the guar seed that I am working from the last 5 years over the enhancing the utilization of these guar seed. Why actually I am working on these guar seed? Because it is a drought tolerant crop, require very less water, very good source of the protein into it. So we can isolate this protein also and can have several applications. Okay, and since we need to look for the sources which are from the food security aspects also, which require minimal resources for their growth and can have beneficial application. So what I am working upon is utilizing this guar seed flour using the non-thermal technology like cold plasma, superheated steam, and the microwave, reducing these factors. Another way is I am isolating the protein from these one, and then we are modifying these protein by the cold plasma or superheated steam, followed by the application into the various kind of the products like ice cream, dairy analog, or as an emulsifiers or as an foaming agent. So in mostly all my studies, this is how we did it. We take the guar seed, convert it into the flour, given the treatment, which could be cold plasma, which could be superheated steam, which could be germination or ultrasonication, and then further analyzing the proximate analysis, effect on functional properties, effect on digestibility, because ultimately how easily they are digestible to our body, it is very, very important. How effectively we have reduced these anti-nutritional factors can be seen into the in vitro protein digestibility also. Different thermal and pasting properties also. So why cold plasma? It's as we all know, it's a non-thermal technology and green technology, and we do not require the presence of some of the solvents to perform. And the temperature is also not rise. It's very less at around less than 60 degrees Celsius. So the nutritional retention would be high, and it has been effective as a surface treatment, can be effectively reduce the enzyme activity and other functional properties. So that's why we did into the current work, and we studied the effect of this cold plasma on the anti-nutritional and nutritional factor, and how effective it was to retain this one. And this is the work we published in the Food Research International over the guar seed flour. Then in another study, we combine the cold plasma with the germination effect. Okay, sometimes germination take longer time. So our target was if it can be effectively reduce this and how it can accelerate and have better retention, so that sometime during the germination process, some bad flavor and no flavors are being generated. In order to avoid that, we combine the cold plasma with the germination. Germination, I hope I don't need to elaborate because it's a very common process that we use it for reduction of the anti-nutritional factor. So for the same effect, what we combine the cold plasma as a pre-treatment on the germination, and then we published this one recently into the Legume Science Journal and see the how it is affecting to the these parameters. Then superheated steam, it is also one of the innovative technology, and I have been working over this technology in the from the let's say past 7 to 8 years. And so we thought upon, since it is a high temperature but a short time treatment, how effective it would be to reduce these factors. So simply superheated steam is the steam that is being generated. If we add additional heat into this one, it can be an effective one with high transfer coefficient to reduce this factor. Why we need to do it? Since it is more energy efficient, does not emit anything into the environment, and because of the absence of the oxygen into this environmental chamber, it is reducing the non-enzymatic or enzymatic reaction. So that's why we use it. And the same work over the guar seed, how it is effective in reducing these factors and maintaining the nutritional quality. We published into the Innovative Food Science and Emerging Technology Journal. I have shared over here the QR code also. People can scan it and can directly get the paper from over here itself with all the details that is required. Another study where we studied the effect of the microwave treatment, because it is very quick process and is effective and lower energy requirements are there. So that's why we studied it. And for the same parameters, and we want to see how effective this one is, and we found it and we published it into the Food Science Journal of the IFT. Another way, so since soaking is very, very commonly used process to reduce, but all the factors are not reduced. So we combine the soaking effect in order to reduce the soaking effect. Because in case of pulses may take 10 hours. So our target over here was how if we can combine it and can reduce the overall soaking time, so that whatever the off flavor compound because of the fermentation can happen, that was we targeted to reduce. And then the same work we published the combining of the soaking and ultrasonication treatment into the LWT journal. While since this seed has a lot of the protein into it, so we thought upon to isolate this one so that can have various application into as a protein source or as a dairy analog or for the fortification purpose. So we since no work was done over this one, so we varied the pH for the precipitation of the proteins at different pH, and then we published it into the food chemistry. But sometimes what happened, these proteins, they have some limited solubility. In order to enhance those solubility and to enhance further functional properties, we modified it using the superheated steam and at a very small interval, so that can have many beneficial aspects. Just today or 2 days before this paper got published into Food Hydrocolloids, and it was a very effective, and a lot of the effort have been put by my team to see the effect and get a aspect onto the property, and we kind of tried to give a mechanism also how it was responsible. So from the key points over the studies that we have done over the guar seed, all the technology, they have their own beneficial aspects and limitations. So we have to see as a manufacturer what kind of the things we require, whether we require it from the seed removal from seed coat or from the cotyledons, or how much percentage reduction we require, or are we looking at what aspects of this one, or energy is important factor. Based upon that, we can go for. So this is my team, because my students are, I am the one directing them, but actually they are the one who are performing the work. So I would like to thank my team for putting the effort to get this so much work done and coming up with outcomes of the research. Thank you, everyone. If you have any queries, please feel free to drop me the message. And I hope I have covered it on time and taken exactly 15 minutes. I'm open to any questions, sessions, if it is there.
Thank you, ma'am, for such an overwhelming presentation. I request Dr. Deshmukh to kindly conclude the session. Sorry. 1 second, please. 1 second. So, after going through the profile of Dr. Deshmukh, I think we are fortunate that he's with us today. So, I request him to share his presentation or his expertise or any kind of work with our scholars. I think they'll be very much impressed by him. Yes. So, please, if any presentation you can give. Okay, no problem, sir. And Mike, yes, sir.
Thank you very much for such a wonderful presentation. And you really did a very extensive work. I enjoyed watching all your different studies with using microwave or plasma. And basically, my group is using genetic approaches to the nutrient component in soybean other crops. So here specifically, I wanted to know how we can integrate this cold plasma, whether in nutrition or processing system or in the lab, whether we have any custom instruments for that or how we produce the cold plasma.
Is there any question? Because I am not able to hear clearly. The voice is little low.
The question is that how do you produce cold plasma? Whether there is a commercially available
Sir, I am still able to hear only few words. Like I heard only the commercial, but could not hear anything. Please, yeah.
Krishna?
Yes, sir. Now I can hear you, sir.
Okay, sir.
Yes.
Actually, congratulations for such wonderful work.
Thank you so much, sir.
I wanted to know how you produced the cold plasma, whether there is any commercially available instrument for that? Or
Yes, sir.
Custom?
Yes, sir. We have a commercial available this apparatus, and we have procured it from vendor into and then Mishra, and he's a CFTRI alumni. And if you need any detail about the model and anything, I'll be very happy to share, sir.
Okay. Or is that is the possibility that we can use that cold plasma system along with the thresher or harvester, so that when I thresh the seed out of the pods, that will get treated?
That can be done, sir, into a continuous mode in order to enhance the capability or the germination capacity of the flow. Again, it depends whether through the plasma we want to enhance only the germination capacity to sow it further and to yield, or we want to check it into the flow. Here we have done it over the flow, but this cold plasma is also very effective if before germinating the seeds, we can give this treatment in order to have a better yield of the crop also.
Okay. And have you tried combination of cold plasma with microwave?
No, sir. We haven't tried with the microwave because microwave itself is also a very energy consuming one, and it's very quick process. And because both are high energy dense product, and if we combine those treatment, I think so.
That will burn out.
Yes. But it will give roasting effect rather than the processing effect.
Yeah. Thank you. Thank you very much.
Thanks. Okay, thank you everyone. I would like to thank all the organizer for giving me this wonderful opportunity that I was able to share my thoughts. And thank you, Modi sir. Thank you, Monica ma'am, Renu ma'am. I hope all the wonderful people over there, just skipping the name, Loveleen ma'am, Sakshi ma'am, Meena ma'am. Thank you so much, everyone. Yes. Shall I take the leave, sir?
Thank you, ma'am, for such an overwhelming presentation. It would be good, sir, that you remain with us for this session, since Dr. Deshmukh will be presenting one of his work to our participants.
Yeah, thank you. Sure, I'll be there. Yeah.
Madam.
मुझे देखो ना करप्ट है
सर ये फाइल आपकी करप्ट है क्या?
वही तो कह रहा हूँ हाँ
इसमें इंटरनेट आ रहा है क्या? सर नेट से डायरेक्ट
डाउनलोड करते डाउनलोड कर दीजिए मैं कर दूंगा करप्ट है ना फाइनेंस यूज
हम मेरे को मेल कर दीजिए आपके मेल पे है
हाँ मेल पे
मेरे को मेल कर दीजिए किसी पे कर दो अनपब्लिश डेटा है ना वो शेयर मेल मेरे को सेंड कर देंगे मेल बना देंगे मेल में टॉपिक मेल
इस पे रिसीव
अनपब्लिश डेटा है कोई बात नहीं
आईडी ना इसमें से किसी को नहीं मिलता कोई भी अच्छा नहीं इससे किसी को अलाउ नहीं होता कल को बता पर मैं सारे स्पीकर पास में ना तो सब इको बहुत ज्यादा मारने लगो
अच्छा
हमारे दो
हमें ठीक है ड्राइव दिखा दीजिए सर आ गया
कल तो नहीं नहीं कोई बात नहीं रोटी भी तो वो पॉइंट कर okay. Actually, I will not take much time. And in this presentation, I will show you how my lab is using genome editing to improve the nutritional quality of food. Actually, the title is crop improvement, but basically we are trying to improve nutrition quality in tomato. So genome editing is a relatively new field, and it is very versatile that provides us opportunity to make any genetic changes. Like in previous talk, we have seen that how Dr. Rashna used different physical treatments to reduce the anti-nutrient compounds in pulses. And if we use genetic approaches to reduce this compound, that will be more sustainable. Okay. So I will just skip. Okay. So I am from Central University of Haryana. This is our campus, and one of the academic block, my lab is present somewhere here. Okay, pointer is not working. हाँ इसमें नहीं होगा ठीक है Okay. So in one of the academic block at ground floor, we started new genome editing lab. So I will skip all the details of genome editing because most of the students are from food technology. So we will directly go to how we use genome editing to improve the nutritional quality. Just one slide for the genome editing. Genome editing is developed using the basic fundamental technique process present in a bacterial genome. Actually, in bacteria, there are several continuous repeats are present, and these repeats help bacteria to have immunity against the bacteriophages. So here you can see this is the whenever bacteriophage infect the bacterial cell, bacterial system will chop down their genome, and some of the small pieces get integrated in between the two repeats, and this is act as a memory. And once this small piece from bacteriophage, that virus, integrated into the bacterial genome, that will transcribe in a long RNA, and then that will get chopped into small pieces. And whenever there is second time infection of same bacteriophage, this memory that will recognize this bacteriophage, that will go and bind to that complementary sequence, and then this Cas9 will make a complex, and that will create a double-stranded break. So this is the simple method how bacteria get immunity against the bacteriophages. And then we use this basic thing to translate into a technology. Okay, so I will skip all the details of genome editing, and then we can start with here. So this is work from my first PhD student, Sanskriti works. He used multiplex genome editing to improve the tomato nutritional quality. So in tomato is a worldwide grown crop. It is major crop for almost all the continent in the world. So every country they produce tomato, whether the weather is good or bad. It is grown in field, it is grown in greenhouses. So a lot of tomato produced every year, but that has a several problems. One of the major problem is the shelf life of tomato. Tomato has very short shelf life. That's why we cannot store tomato for a longer period. And therefore, in the market, the tomato price always fluctuates. Sometimes it is 2 rupees kg, sometimes it reaches 200 rupees kg. That's why it is very important to improve the shelf life of tomato. So, in the Sanskriti's PhD program, we selected 3 different genes in tomato to make a genome editing. First one is the SP5 gene, that regulate plant architecture. Whenever we knock out these genes, that will change the plant architecture of tomato. Second gene is the pectate lyase. If we knock out the pectate lyase, that will improve the pectin content, and the fruit shelf life will be improved. And the third one is a lycopene beta cyclase. If we knock out lycopene beta cyclase, that will improve the lycopene. Okay, so this is a carotenoid biosynthesis pathway. So here this lycopene beta cyclase work. Whenever there is conversion of lycopene to beta carotene, that conversion happens because of the lycopene beta cyclase. Okay, so this is lycopene, and that get converted into beta carotene because of the lycopene beta cyclase. And if we knock out this lycopene beta cyclase, that will improve the lycopene content. And lycopene is a compound that gives red color to the tomato. So many times I ask this question: if we stop the conversion of lycopene to beta carotene, we are getting better red color. But in most of the cases, we wanted to have better beta carotene because we are producing golden rice. We are trying to make a golden banana, because in golden rice or golden banana, that will have more beta carotene, and that is a vitamin A precursor. But in this case, I am targeting lycopene beta cyclase to stop the conversion of lycopene to beta carotene. Then the question arises whether if we are getting more red color of food, whether that will be equally healthy or not. So this is also healthy because lycopene is also a nutraceutical compound, which is helpful for the health. And for beta carotene, we have several different sources like papaya, carrot, mango. But for lycopene, we have very few sources, and tomato is one of that. Therefore, it is good idea to stop the conversion of lycopene to beta carotene to improve the lycopene content. And if the lycopene improves, that improves the red color of tomato, so that finally we will get the better market price to the tomato. So this is genome editing is very simple. First, we design a construct, plasmid construct having Cas9 protein that is that actually do the double-stranded DNA break, and the guide RNA. Guide RNA is the whichever gene we wanted to target for the genome editing, we just take a 20 nucleotide complementary sequence of that, and that we use in this system. So once the construct is ready, then we deliver that to the plant. So for delivery, we have several different methods like agrobacterium mediated transformation or gene gun. So in tomato rice, we usually use agrobacterium mediated method. So we deliver this to the tomato plant, and once transgenic ready, then we sequence the target size. Suppose in this case, if I am targeting Lycopene beta cyclis, I need to sequence the Lycopene beta cyclis target site and then see whether there is any changes or not. So if there are desirable changes, then we can develop that line into a variety. So this is a multi-target system where we can use multiple guide RNA. Multiple guide RNA means if I wanted to target 4 different genes simultaneously, I will use only one promoter, then guide RNA 1, guide RNA 2, guide RNA 3, guide RNA 4, 4 different guide RNA for 4 different genes. So that allows us to target multiple genes simultaneously. Okay, so for selection of the target size, there are already so many online software tools are available. We can use that tool to select any specific target site in our gene, the candidate gene. And then we can clone that guide RNA in our plasmid vector, and then we can use that for the transformation. So in tomato, in our case, we use Pusa ruby variety, tomato variety. It is widely being used in India in different labs for the transformation. Earlier, it was thought that this tomato Pusa ruby is very easy for transformation, but later on, we developed a protocol that we can use for several different tomato varieties for the transformation. And then here we use the RNA sequencing data for Pusa ruby variety to select confirm the target site. And then the single target construct we developed. Here we have promoter and then Cas9 gene. So this promoter will transcribe this Cas9, and another promoter that will transcribe the single guide RNA. So this is more technical, but just we will wrap up it quickly. So for multi-target genome editing, we use golden gate assembly method. That is basically cloning method where that allows us to clone multiple fragments simultaneously. So when we wanted to target 3 different genes simultaneously, we need to use a golden gate so that the 3 different guide RNA can be cloned in a single vector quickly. And then we used optimizer protocol, which we use for the Pusa ruby, Arcavicas, Pusa Ridop. These are the tomato varieties. Pusa ruby is a variety which is usually used in the experimental purpose. This is not being cultivated in a farmer's field. But Arcavicas, that is being cultivated in the farmer's field. Pusa Ridop is also there. Solanum peruanum, this is a wild type tomato. So we develop such a protocol which we can use for all different type of tomato varieties as well as the wild type tomato. So this is the steps for the tomato transformation. We usually take a hypocotyle, then we co-cultivate that with the agrobacterium. Then there is a step of shoot induction, then root induction, and finally in a 6 to 8 week we can get a transgenic plant. So once we have a transgenic plant, then we sequence the target site to see whether there is any editing happens. So here you can see when we sequence this with Sanger sequencing, there are several bases deleted. So even if any gene, if there is only one base per deletion in a coding region, that will make a frame shift. That means the protein will be completely changed. Once protein change completely, that means that will no longer functional, and then we can achieve the desirable change. Like in case of Lycopene beta cyclis, if that protein become non-functional, that will stop conversion of lycopene to beta carotene, and automatically the lycopene content will improve. That's how we use here genome editing to improve the lycopene content. So these are the results. T0 means the whatever plant we get first time from the transgenic, that is T0 stage, like F1, F2. This is in transgenic. In case of transgenic, we use the terminology T0, T1, T2. So in T0 plant, we achieved plant with the 11 base peg deletion, 5 base, 17 base, 10 base. Here nomenclature is like AE is Arcavicas, variety 64 number of plant. TR is a Pusa ruby variety, 38 number of plant. TED is a Pusa Ridop. This is SP is a Solanum perennium. So these are in all different varieties we achieved genome editing for this pectic layers. Like one beta cyclase, then this is pectic layer, this is SP5. All 3 different genes in 4 different varieties, we developed a genome edited plants. So these are the result how we achieved it in a T1 stage, T2 stage. And then these are the phenotypes. So here you can see this is Pusa ruby, a wild type. When we edited SP5G in Pusa ruby, that plant becomes such a short one. So this tall plant becomes only 1/3 of the size of wild type. Then question arises when we make it compress, whether the production will be reduced or not. And even by compressing the size of tomato plant, the production will be same. And the benefit is that this plant usually take 5 to 6 months to complete the crop, but this will take only 3 to 4 months. So we get advantage of having short period of cultivation. And in 4 different variety, if we knock out the gene, that will give a 4 different type of phenotype also. That's why we selected diverse variety. So here you can see this is Pusa ruby. In case of indeterminate tomato, this Pusa ruby is an indeterminate type of tomato. There is typical pattern of flowering. So there will be flower, then 1, 2, 3 leaves, and then again flower. So every after 3 leaves, there will be flowering. But when we make a knockout of this SP5G, there is a flower, and then one interleaf, second leaf, and then flower. So that's how the flowering in alternative internal, the total number of flower will be the same even the plant is compressed. So ultimately, the number of fruit per plant will be the same. Here, this is Pusa early drop. In Pusa early drop, there is a flowering of every after single internode, like flower, then internode, then flower. But when we did the same SP5G, here the flowering is at every internode. So same gene can give a different effect in a different genetic background. So here you can see this is for the pectin layers. This is wild type, the above rows, and the bottom at the bottom this is genome edited. So when we edited this pectin layer gene, that improve the pectin content in pericarp, and that's why the shelf life is prolonged. So here you can see this is completely wrinkled, but this is still fresh one. So we can improve like 2 to 3 weeks of shelf life in a storage condition. And in tomato, whenever we transport tomato from one place to another place, that also get damaged during the transportation. But because of the higher pectin content, the damaging also reduced. So here you can see the fruit get damaged. But in the wild type, the damaging is much more higher compared to the genome edited tomatoes. Okay, so for lycopene content, when we edited that gene, lycopene also improved, then the fruit become more red in color. For root trait, this is not yet well characterized, but in wild type, the root length is this much, but in our edited plant, you can see the root is 3 times more. If you compare the shoot, this is exactly same in all the 3 edited line as well as wild type. The root length improved 3 times because of the genome editing. Okay, now we can start the next topic, that is moving toward the food as a medicine. Tomato provides us excellent opportunity to consider it as a food as a medicine because we consume tomato as a raw fruit. We consume that in a salad. We can consume tomato as a processed food like ketchups and sauces. We consume tomato as a cooked food. So in all different forms, we can consume tomato. Suppose we improve any nutraceutical compound in tomato, and that is not heat liable. In that case, we can consume tomato directly as a raw food. So that this has an opportunity for us to convert the food as a medicine, and that will give us sustainable benefits to our health system. So in that case, we are targeting GABA production in tomato. This studies, similar studies already published, and in Japan, GABA-rich tomato is already in the market. So we are just replicating the same study with the Indian cultivar. So in this case, we are targeting this GAD3 gene that has autoinhibitory domain. If we remove this autoinhibitory domain, that will improve the GABA content in tomato. And GABA is very important for India because in India we have a lot of patients with the BP, that hypertension, and GABA provides benefits against that. So that is very important for us. Second thing, that vitamin D deficiency. Almost 50% of the people, whenever get their blood tested, they will always see that they have vitamin D deficiency. That is also a big challenge for us. And in tomato, we are also trying to improve vitamin D by targeting this SL7DR2 gene. If we knock out this gene, that will improve the vitamin D in a tomato. So this slide is also very important because most of the time, whenever you start thinking about genome editing and transgenic, there is always concern that may need a very big molecular biology lab, and it is very difficult for to think about to starting a molecular biology lab. But this slide shows that in University of Nebraska, they provide paper facility. That means even if you provide $10, that is around 1,000 rupees, they can give a vector construction. If you give $7.5, they will give tobacco transgenic plant 10 plant for $7.5 only. For $3.5, they can give RFW transformation. So it is, if similar facility we develop in India, there will be no need for breeders or people who are working in the food science to think about to starting the lab for genome editing. They can just get it done with the commercial facilities, and then they can work on the product. Okay, so similarly, we have several different projects with genome editing. In one of the projects, we are trying to reduce lactose activity in rice, and we are also trying to knock out this fat to that stop conversion of oleic to linoleic, so that there will be more oleic acid. In another project, we are trying to make a male sterile improved rice hybrid system by targeting two different male sterility systems. Okay, I will just skip all those different projects. And okay, so the tomato work is from the Sanskriti's PhD program. She is presently doing cold-back camping in Denmark. Yogesh is my another PhD student. So he worked with the aquaporin dynamics in submitted PhD. These are my PhD students who already passed out, and they worked with different aquaporin projects. Presently, I have this sixth student, Mukesh Badal is here. He's working with the vitamin D and GABA improvement in tomato. Pragati and Pawan, they are working with the different projects. These are the new two students. Thank you very much. If you have any question, you can ask.
Just I want to ask one question. That is what I came across through industrial interaction, especially with the tomato case. Yes, as a consumer or a general observation for the food processing and all, that what tomato we are getting, it is a very healthy and with extended shelf life, as you shown that the lycopene content is increased, and of course they are very healthy, looks like very, means they have a extended shelf life. But what industry people, what I was interacting, that they see there is a shift in the pH of the pulp. One, that is, of course, they said that okay, that we can make up with the addition of some acid and all. But the drawback, what he highlighted, that the flavor part, the aroma, what we get from the tomato original, that is totally lost in this. Though lycopene is more, looks very healthy, very bright red color, attractive as a consumer plate or as a industry also, we are very happy because we are getting more pulp, more a bit little total solid has increased. All those factors are okay, but we are at a loss of the flavor.
Yes.
And that flavor is that what tomato flavor, that acidic flavor, what exactly we need. That is very important for sauce, tomato sauce or ketchup, what they are manufacturing. That we can see with the use of artificial flavor in our sauce to compensate with that. So just I want to know your with this.
Actually, the tomato breeding or any breeding program, that is driven with the market requirements. So initially, when people were having more desi type of tomato in a market, that time consumer preference was for the tomato which is a firm, having less seed. And then automatically breeders started producing more varieties which are like more firm in shape and having less seed. And then again the consumer preference shifted. Now consumer want the tomato more like a desi type which has a more seed. And then breeder also started making new varieties with the same background. So in case of tomato, it is like whenever we think about transgenic, always people get in some notion that is taking something original out of that, and whatever will the transgenic will deliver, that is not a natural thing. But in case of genome editing, that is the different with the transgenic and genome editing. In case of genome editing, we are not adding any gene. We are just modifying the whatever genes are already there. And second thing, that we are trying to modify only those genes which are not naturally already present there. Like pectidase, if we wanted to knock out the pectidase, if pectidase already, there are varieties where we don't have pectidase functional. There are already varieties where we don't have lycopene beta cyclase active that has a very red color. These things are naturally. But if we use breeding approaches to combine all different desirable genes in a one genetic background, that will take a lot of time. That's why we are here using the multiplex genome editing, where we can use multiple different genes simultaneously in a segmenting background. But here we are not delivering something which is not present in a nature. So all three different genes, their mutations, the editing that is already present in a natural tomato variety. So that care we usually take, because if we go with this system, like considering the natural germplasm, then only we can deliver the product in the market. Otherwise, there will be again the same thing that the consumer will think that this is something artificial and I don't want. I need something that is this type. So we always keep that thing in our mind whenever we design any genome editing program. So for GABA content, if you are getting tomato with a high GABA and if you already have a blood pressure issue, then you will definitely, the consumer will definitely go for the GABA rich tomato, because that is giving something essential for your body. So that's type of, we first chalk out all these such issues and also consider the consumer's preference, what will be there in after 10 years, because whenever we start with such a project, that we can complete that in a 1 or 2 year, but to get that product in the market, that definitely get the 5 to 10 years time. So that we need to advancedly think about that, what will be happen, what consumer will prefer after 10 years. Thank you. Okay, I need to conclude also.
It's a very nice presentation. I have a very small query actually. I think there's so many varieties in the fruits which are perishable one. So like we are doing for the tomato, can we do for there also like gene mutating and all? Because I think we're working with the same gene. There's some alteration in the gene we are doing.
Yes.
So can we do for different varieties of other fruits also? Like
yes, yeah.
Perishability is high.
Genome editing we can use in many different crops, many different varieties. Like some of the project we have with the rice, where we wanted to improve the brown oil. Rice brown oil already there in the market. Okay. So we wanted to improve the brown oil. Second, we are also working with the soybean, because soybean has a very typical pungency, different taste. Like Indian people don't like that soybean flavor. We eat soybean a lot of with the soybean nuts and those things, but it is not till we cannot eat soybean as directly like other pulses, because that has that beany flavor, and KT has a one anti-nutrient also. So we are targeting those to remove the anti-nutrient and to improve the flavor in soybean. And there also we are making it. There are some varieties already there. So we are mimicking whatever already naturally present in a nature. Just we wanted to have those same trait in a variety which gives a more production.
Thank you. Thank you.
Anybody else wants to any question if you have? Anybody? So thank you so much, sir, for your wonderful presentation. It is always a lifelong opportunity for, especially for me, to hear you and your findings. So I have a very it's not a question, it's just a curiosity that if we are going to edit something in the natural products, so how we can consider it that it's not edited means it is a natural, because if you are decreasing or increasing any components in the natural tomato or rice or anything else, so how we can consider it that it is a natural? Why we people like you are saying that if anybody have some blood pressure issues, then he or she should consider the GABA rich tomatoes. So why human beings, they are not working towards the natural life, stressful life, instead of taking some medicine, some GABA rich or protein rich or vitamin rich fruits? Because from the very beginning, from the evolution time, each and every products are naturally present. Now we are starting to edit into the gene system, into to increase the protein, to increase the something else. So how we can consider these products are very natural, and we should consider it if we are going to edit somewhere, means the genes or anything else.
Okay. Actually, in tomato, there are natural varieties which has a high GABA content. But if some varieties are in China, their preference will be different. Those will not give a similar production in India. So if we exactly make the gene which is already naturally present in that GABA-rich tomato variety in Indian variety, so we are not creating anything new, because there are already varieties which give a higher GABA content in tomato. Just we are bringing that trait in an Indian variety, so that we will also have the higher production as well as a high GABA content. Another simple example is rice. In case of rice, we consume aromatic rice like a basmati. So there is one gene that is bad H2 gene. If there is 8 base pair deletion in that gene becomes non-functional and that gives aroma. So whatever aromatic variety you consume, in every variety, that gene is not functional.
Okay.
Now, if I wanted to convert a variety which is being grown in Odisha, which gives very high production in Odisha. So traditionally, we used to cross that variety with the aromatic variety, and then we make that aromatic. But whenever we cross, that take a time, and that will not keep the exactly same productivity of the variety. We call that as an unwanted genetic linkage drag. But if we use genome editing to make 8 base pair of deletion in variety which is being grown in Orissa, that will convert that variety into aromatic variety.
Okay.
Now the 8 base pair deletion we created, but naturally that 8 base pair deletion is present in all the aromatic varieties. That's why I'm calling it that whenever we are using genome editing, we are keeping the product naturally as that is in a different varieties.
Okay. So thank you so much, sir. Thank you. Thank you, sir.
Thank you.
Thank you, sir, for such an overwhelming presentation and sharing your knowledge with our participants and students. I request our HOS, sir, Monica ma'am, Durgesh sir, to kindly felicitate our session chair, Dr. Rupesh Deshmukh sir, with a memento. I request Renu ma'am also to please join us on stage. Sir, please. Thank you, sir. I request our dignitaries to be remain on the stage and kindly felicitate Mr. A.K. Bhatnagar, sir, with a memento. Sir, I request Rupesh sir to be on stage, sir. Thank you, speakers. Thank you for joining for the 8th AMIFOST 2025 and for your knowledge sharing. Our next session, technical session 5, food safety and quality, would be an online session, and our speakers has joined us online. I may request our dignitaries to kindly proceed for our networking team and the participants as well. The session will remain online. You may join through the link which has been provided in the program schedule. So we have been joined with our session chair, Mr. S.S. Raghav, Director, National Food Laboratory, Ghaziabad, India. Sir has distinguished experience of 34 years in the field of food safety, particularly in food analysis by chemical, instrumental, and microbial methods in the food matrices, that is, milk and milk products, fruit and vegetable products, cereal and pulses, spices and condiments, packaged drinking water, mineral water, nutraceuticals, sweet and confectionery, alcoholic beverages. In his professional career, Sir has prepared manual of method of analysis for food from 2004 to 2007, preparedness of scientific technical aspects for expression of Government of India point of view in various national and international platforms, also represented National Food Laboratory Ghaziabad at various meetings of Codex Alimentarius Commission. Sir has delivered lectures, presentations, and laboratory demonstrations on food chemistry, sampling, laboratory accreditations, fruits and vegetable product analysis, food microbiology, concept instrumental analysis. Sir has also worked with Central Food Laboratory, Kolkata as Chief Technical Officer and worked with National Food Laboratory, Ghaziabad as Chief Technical Officer and Joint Director. Thank you, sir, for joining us online.
Good afternoon, madam. Can I have my other session member, please?
Session.
So no one is there. I hope everyone is joining online.
Yes, sir. So
All the others are online. I understand.
Sir, Peterson is already there for this session. He has joined us online only.
Okay, it's me only.
So Dr. Peter is there.
Dr. Preetham.
Peter, sir, from Spain. Peter.
Okay. Voice is little breaking. Am I audible to all?
Yes, sir. Very much audible, sir.
Okay. So good afternoon to all. At the first, I want to introduce myself. I am working as a director of Food Safety and Standard Authority of India, and I'm working at National Food Laboratory presently, which is an state-of-the-art pioneer food testing laboratory of Food Safety and Standard Authority of India. When we talk about the food quality and food quality assurance in India and food testing ecosystem in India, Food Safety and Standard Authority of India is a pioneer organization which has come into force in 2009 after implementation of Food Safety and Standard Act. As all would be knowing that Food Safety and Standard Authority of India is a prime nodal agency for controlling all the food standards and aligning our standards to the national and international standards, which are prerequisites of WTO. I hope everyone knows the word trade organization, and it has created a benchmark. There are two things, which is SPS and TBT. TBT states that we should eradicate our trade barriers to the trades. So, for minimizing, we have to harmonize our standard. We have to harmonize our food testing ecosystem with the international level. So, international level is nothing but daily emerging trend of the sophistication. So I start with a little introduction of Food Safety and Standard Authority of India. This is authority which is, as I told you, is a pioneer authority for the ensuring safe and nutritious food to the citizen of India, and it is having food testing, a great food testing ecosystem. We have close to 250 food testing laboratories across India, which are notified by the Food Safety and Standard Authority of India. And primarily, they are accredited by the NABL, which is an which creates a level playing field where every lab is required to pass their exams. Like they have to be assessed by their auditors, and after accreditation by the NABL, FSSAI recognized them as a food testing laboratory. In fact, it is written in an act that if a lab is not accredited by the NABL, it is not a food testing laboratory. So we have close to 250 food testing laboratory across India, which include government, semi-government, CSIR, ICMR, ICAR labs, and so for the FSSI on laboratory network is concerned, we have four premier food testing laboratories in India, which is one is at this place, which is very near to your place. It's in Ghaziabad, and one is at Mumbai, one is at Chennai, and one is at Calcutta. So we have a lot of sophisticated equipments. See, food is becoming more and more smarter for the profiteering, or the point of view of the profiteering, that people want to earn more and more profits out of the food, and they want to play with the quality. They want to put some adulterations in that. Adulteration is an old word now. We call it the safe and the unsafe. The basic difference between safe and unsafe is once it is contaminated, it has contaminants like it has metal contaminants like lead, arsenic, cadmium, mercury. When it has the pesticides, like so many pesticides are there. we have close to 213 pesticide regulated in our regulations. And when we have the aflatoxins, when we have the microbial contaminants, food is called the unsafe. When it is deviating from the standards, it is called the substandards. Deviating from the standard means if the food is required to have say 50% of the protein, if it is not having that much quantity of the protein, so then we call it substandard. And there is another category which is a misbranding, which is you see a lot of government is doing a lot of promotions for making people aware regarding the label declaration so that the consumers can be well informed. So from that perspective, there are certain regulations which governs the how a label should be put on what are the details which a food package item is required to carry. So when that is not there, that is not in the way it is required to be there, and when quality of the some quality characteristics like fat, protein, carbohydrates, nutrients, if they are not in their required quality or not matching to the label declaration, it is called the substandard. And when it is having contaminants like those I told you, pesticide, metallic contaminants, aflatoxins, ochratoxins, and microbial toxins, whether they are hygiene or whether they are pathogens, so then it is called the unsafe product. So for ascertaining a product whether it is safe and fit for the human consumption, we have 4 national food laboratory. Beside this, we have authorized close to 200 labs of the private organizations. They are working across the country, and they have the very broader and very important role to play in the food safety, maintaining the food safety ecosystem. So they ensure that the food is tested for the government. They ensure the food is tested in a international as per the international requirement for the state governments, and they ensure the food is also tested as per the requirement of the industry. So for ease of availability, it is distributed in whole of the India. Beside this, we have CSIR and ICR, certain CSIR and ICR labs, which are there, which is having, if they have the mandate of food testing, they are also authorized by the Food Safety and Standard Authority of India. So, so far the food adulteration is concerned, know that people always try to maximize their profit by reducing the quality of the food, making it contaminated because of using less stringent quality policies and like that. So for that purposes, these food testing ecosystem is is required. Beside this, Food Safety and Standard Authority of India has started Eat Right India campaign, which is an offshoot of the campaigns which are mandated by the Prime Minister in 2017, which is the Ayushman Bharat, Poshan Abhiyan, Anemia Mukt Bharat, and Swachh Bharat Mission. So, in the Eat Right India movement, we do a lot of we take a lot of initiative for countries' food system in order to ensure safe, healthy, and sustainable food for all India. Through Eat Right India movement, the tagline "Sahi Bhojan, Behtar Jeevan" thus forms the foundation of this movement. Eat Right India adopts a judicious mix of regulatory, capacity building, collaborative, and empowerment approaches to ensure that our food is good, both for the people and the planet. Further, it builds on a collective action of all stakeholders, the government, food business operators, civil society organization, experts and professionals, development agencies and citizens at large. So there are a lot of initiatives. We organize a lot of Eat Right Melas where we invite vendors, where we invite the peoples so that they can get the knowledge of how the government is working, how the food is ensured, what are the regulations, and we have also identified certain Eat Right streets where we identify the area where maximum food business operator of small sizes are there, like like street food hub, eat right street is, those small vendors with the vending machines and vending telas and all, moving vehicles and small shops. There we tell them how they can make their food more safe, how they can make people assured about the safety of their food products, how they ensure the quality of the ingredient of the raw material which they purchase from the local market, and what are the basic hygiene purposes. We ensure that every vendor is having a proper sanitation and disposal mechanism, and they are taught about how to handle the food more in a more careful way, so that it becomes safe to the public at large. And we also do so many initiatives, like we organize so many sports events also, where we invite prominent sports personalities, where they can where they can where they encourage people to live healthy and eat healthy life. So I coming back to the lab services. So all the labs nowadays are equipped with the sophisticated equipments. So most of the equipments are they are capable of analyzing the food contaminants in ppm and ppb level, and they are generally lcms, gcms, hplcs, icpms, icoa, icp, oes, and various kind of chromatographic equipments, nmr, and we have the all the national food laboratories. We have all the state of the art microbial testing laboratory, where we can do all the hygiene parameters like yeast and mold, TPCs, and pathogens. Almost all the pathogens which are given in the regulations are covered in that. So in that way, we ensure that the food which is sold in the Indian market is generally safe. We have a mechanism of collecting food samples. We have the regional offices. We have 4 regional offices across India, which is north, south, east, and west, where we have the food safety officers and our office and other officers. Those who go to the field, go to the shops, go to the malls, go to the small selling vendors. They collect the food samples and it is tested, and then we would do a lot of surveillance. We do close to 15-16 surveillance in a year, where we collect the samples from the general market. We collect the sample from the food industries, from their processing lines, and we analyze them critically, and we evaluate what are the pattern. We draw a pattern which food is having maximum number of the adulteration, so that we can focus our drive. We can take more of the regulatory samples from those area, and we can analyze them more critically. And there is a mechanism, not only the analysis. The story doesn't end at the analysis and making the reports available to the regulatory authorities. It starts with this. It starts with the analysis. If the sample doesn't conform to the required regulation, then a prosecution is launched. If for the surveillance, at the first place, we identify the area, we identify the food matrices, which is more prone to the adulteration, which is, where the most of the compromises are there. And then we focus on those areas, then we do a rigorous sampling, and then we analyze, and then our FSOs and our officers, they prosecute them. They penalize them. There are defined fines in our regulations, which is imposed on them, and they are stopped for doing business. They are stopped producing further items in their factories. So it's like that. And then there is also a mechanism of getting the second testing, which is the referral food laboratories. For that purposes also, we have established close to 22 food testing laboratories. We have assigned them the referral food testing work, which is a secondary food testing. Secondary food testing is more rigorous and more accurate, I must say. And of course, they are all are NABL accredited and have a level playing capacities. And when the food is finally comes out of the referral food laboratory, if it is confirmed, then it is okay. If it isn't confirmed, then prosecution is launched from the shore, and the responsible business operator is prosecuted. So in that way, our food testing laboratories creating a very important role of creating evidences. It's like generating forensic evidences, and unless until you have some evidence. Some blood sample, like DNA samples, or like some ballistic samples, which matches with the which put the culprit, which proves the culprit is a real offender. It is like the same thing is done by us. We ensure that all the evidences are given to our prosecuting authorities so that they can prosecute the offenders, those who play with the life of the people. So we provide the evidences, we provide them the test reports, we provide them the assessment, which area is requiring more focus. In that way, we are doing. And besides, so far this food testing is concerned, we have close to 4-5. We have close to 18 regulations in FSSI, Food Safety and Standard Authority. In the meantime, I would suggest you to keep on searching the website of Food Safety and Standard Authority of India, which is fssi.gov.in, and you will find a lot of interesting material in that for the people those who are in food sciences in any way, be it the food technology, be it food science, be it nutrition, be it some other way. If it is connected to the food science, definitely people will you all will find something or other of your own choice, of your own interest. So we have, coming back to the regulations, we have close to 18 regulations which are for the various purposes. We have a very transparent system of formulating the regulations, standards like, and we have 4-5 regulations out of those 18 regulations which are of immense use, which are food product standard and food additives, which is a very comprehensive regulation where we talk about all the standards, contaminants, microbiological things, and additives which are allowed in the food. We have the contaminant regulation where we describe in detail the contaminants which are having the MRLs, maximum residue level in the food, which is very critical and very important. So far the analysis is concerned, so far the general awareness is concerned. We have labeling regulation, a separate labeling regulation, which is a very comprehensive regulation, and I request all of you to go through all these regulations. And we have sampling regulations where we define the roles and the responsibilities and the quantity and how the sample is taken. And we have various regulations for how we do the business at Food Safety and Standard Authority of India, which is extremely transparent and address all the businesses which we do, all the work which we do here at FSSI is properly laid down. We have scientific committees, we have scientific panels when before setting any standards, we have a robust mechanism where we ascertain whether a product is having having some scientific attributes, and the additive which is required to be allowed in that product is having scientific necessity. Unless until it is scientifically required, we do not allow any additives. We have in 2015 we have aligned ourselves with Codex regulations, and where we have aligned close to 1500 additives with the Codex Alimentarius, which is an apex body for the setting up of the food standards. So these are the things which we do at the Food Safety and Standard Authority of India to ensure the food quality. So lot of new things are happening. We are emerging day and night. I mean, we have a scheme of the DART where we encourage the small entrepreneurs. And I also, if someone of you have any interest in developing any new technology for DART scheme, which we have in place at the FSSI, you can always search that DART scheme at our website. And there we encourage the young entrepreneurs, small entrepreneurs to come up with the technologies which is useful for the food testing. Particularly, we require technologies which can give the results in a shorter time. See, we also test the lot of import items. For the import items, we require technologies which are short in duration. So when we test the import samples for microbiology, it requires, and the classical method requires, I mean, all 3 to 7 days of the incubation before reaching to a concrete conclusion. We require the technologies which can cut short those time. We have already encouraged. We have already adopted so many technologies which are there. You can go to the website of DART, where we have accepted. We have recognized close to 72 technologies across the India and abroad, which can be useful for reducing the time of the analysis. And we are always open to adopting new technologies. Besides this, we have so many new initiatives where we ensure that the food testing ecosystem in the states. See, in Indian political system, we have states, we have center, where center is always having a better technologies, better system of addressing an issue, addressing it touches, I mean, broader outlines of human life. So we have state of the art equipments. We have state of the art laboratories with the state of the art manpowers also. Where in many states, I mean, this is lacking. So we do a lot of handling there. We provide them assistance with equipments for setting up the laboratory, for setting up the microbiological sections or in their lab, and we provide them the equipment with the support up to 5 years, so that they can run the equipment without any fear of providing support from their own state government. So with this, we ensure that in the whole of India, there is a level playing field for the food business operator, which is ensured by the Food Safety and Standard Authority of India through their State Food Safety Commissionerate. So with this, I like to ask my co-panelists who are there to show the light on their subjects, so that we can also know, the audience can also know about their thoughts.
Thank you, sir, for your insights on food regulations. Today we have a pleasure of hosting a renowned scientist and an exceptional academician and esteemed researcher, Dr. Miguel Prieto Lage, Associate Professor, University of Vigo, Spain. Sir has dedicated his career in the field of additives, natural pigments, green extraction, etc. He has addressed several research topics across different fields, but always keeping food science as his major research area. He has strongly contributed to the optimization, implementation, and development of various extraction and analytical techniques for different natural molecules and to their bioactivity evaluation. Having more than 600 publications, over 10,439 citations, and a remarkable H index of 55, It is truly an honor to have Dr. Prito with us today. His insights and expertise will undoubtedly inspire us and provide us invaluable knowledge.
Great, Mehul, Angel. It's nice that you are here with us. Can you hear us?
Yes. Can you hear me?
Yes.
Yes,
I can hear.
you.
Hello, good morning. Thank you all for the opportunity of welcoming us from the University of Vigo. I would like to apologize because Dr. Miguel is not present today. He had a personal issue. He didn't make it today. So my name is Maria. I'm working with him in the laboratory at the University of Vigo, and he asked me if we can make the presentation anyhow, and to apologize especially with Professor Monica that was hosting the conference and invited us every year, and of course with all the presents and the conference speakers, Dr. Professor Surender Singh. We would like to show our most sincere apologies first of all, and then, if you let me, I would start with the presentation.
Please.
Please confirm me if you can see.
Yes, ma'am.
Yes,
ma'am. It's visible. Please.
And it's
yes,
changing.
ma'am. Yes, ma'am. It's changing as well.
Okay, perfect.
Thank
you very much. So, as we said, we are the nutrition and food group located in Vigo, in the University of Vigo in Spain. And today we would speak about extraction of bioactive phytochemicals from natural sources, optimization and industrial application. The research group is now led by Professor Dr. Miguel Angel, and we belong to the Department of Analytical Chemistry and Food Science in the Agroecology and Food Institute. The presentation would be divided in the following table of contents: from presenting the University of Vigo, from our group, about us, the main work research line, some examples, publications and projects, our work with companies, what we think it's very important, and the contact information in case any of you would like to join us in the future. So our group comes from Vigo, Galicia, in Spain. It's located in the northwest by the sea, as you see in this image. We are in this small part, and the University of Vigo is formed by 3 cities, 3 campuses, which are Ourense, Pontevedra, and Vigo. They are specialized in each topic. The university is relatively young. It was established in 1989, and it's a public university in Spain that has, at this moment, around 20,000 students. And I would like to show you in the following slide some rankings about our university, because we are really proud of being one of the highest university in food science and technology. This year we were 34 position, but last year and the previous we were ranking around the 10th or between the 25 first. We are especially highlighted in food science, but we are also good in oceanography. And as you can see there, you can manage the rankings in Europe, in Spain, in the world. So our group, this is the building where we work every day, is the technological city in the University of Vigo. And this is a brief view about the profile of Professor Miguel Angel, which, as you said, had more than 200 publications. This is the record for the citations in Google Scholars, and he has been working with the University of Vigo and the Research Council in Spain for more than 20 years. We would like to show the position by field to show where is our main expertise. So you can see that one of the things we most highlight is the green extraction techniques, the natural pigments, and then the response surface methodology. This is the way we usually applied our knowledge. Professor Miguel has a strong background in mathematical modeling, and the way he has transferred this knowledge to our field of work related to natural extraction and bioactive compounds is through this tool, response surface methodology. We also have a strong CV in additives. He is ranking second. The first is also a very good friend of him, and for me, I'm there in the 5. So we are trying to make our contribution to this field a lot, and also in dose response, because as I was saying, he has a strong background in mathematical modeling. Regarding the group, the journals and fields we usually publish more are related to agricultural and biological science. And in the right left, you can, in the right part of the slide, sorry, here, you can see the distribution of the journals. We have strong contributions in food chemistry, in Food Research International, foods, functional foods, critical reviews in food science and technology, and most of these articles belong to the first quartile, but more than 22% belong to the first decile, which is also a good mark about the quality of the scientific papers we are developing. Then we would like to highlight some collaborations that we use to complement our work. Because of the geographical proximity, we work a lot with Portugal. They are our most beloved members. We work very close with Polytechnic Institute of Bragança, that is also leading the ranking in the world for food science and technology, with Professor Lilian Barros and also with Professor Isabel Ferreira. We also work with Professor Fatima and Professor Beatriz from Porto. And we would like to highlight, there are not all of the collaborations here, but we work with Professor Magdi Jafari, which is highly cited researcher from Iran, Luigi Lucchini from Italy regarding the metabolomics approach. We have connections in Argentina, also strong collaboration with China, focus on the Shandong Academic Agricultural Science and the Ocean University of Science with very good friends, Professor Kao Li and Ninjan Li. And we have other collaborations, newer collaborations. In particular, I would like to highlight here the work of Professor Tarun Belwal, that I think it's also collaborating with the conference, and it's a good friend. And then I would move to present our research lines. So basically the objectives, we have 2 objectives. One of them is the obtaining extraction rich in bioactive compounds, and the other one is to improve, to optimize the extraction of these compounds by ecological and conventional techniques. So with this idea in mind, we always like to divide the matrices that we use, the raw materials we use in 3. We work a lot with traditional and medicinal plants. We have been working with Asteraceae family. We have been working with Camellia, not only Camellia sinensis from the tea, but also Camellia japonica and Verbascum sinuatum, but a long list of traditional Chinese plants and local medicines. We also work with food industrial waste. We had a big project working with byproducts from the olive oil industry, the wine industry, that are very strong in our country. And at last, one of the topics I like the more is the work in algae and seaweed. We work with several companies in our community. In our case, in Europe, the algae are not so consumed as in Asia, but it's boosting these last years, and we are trying to provide our knowledge in this field. So the research flow, it's always or more often organized like this. First, we focus on the extraction, then the identification of the bioactive compounds at the raw matrix, the study of the biological properties, and then we moved for the final application that we like to focus on food industry, like developing new additives, as I was saying, or complement pharmaceutical industry with some kind of encapsulation system or something similar. And also we'd like to work in the scaling up process, joined together with the companies. However, this is the the research flow, the research line, but most of our work is focused on the three first stages. The final application, sometimes not everything reaches the final application, and for the scaling up, it happens the same. But I would like to focus on the three first. Well, this is again our research group. This is Miguel Angel in the right side of the slide. And for the extraction, we would like to present a little bit what we can do, the facilities we have in our laboratory. We develop extraction techniques by ultrasound, microwave, pressurized liquid, heat-assisted, high-pressure, supercritical fluids, and substrate extraction. We perform the preliminary identification with spectrophotometric methods and purification systems with ultra and nanofiltration. Sorry. Then for identification, we have these methods, HPLC, coupled with different detectors like DAT, but also coupled with mass spectrometry in tandem. And the same happened for the gas chromatography that we use for different applications. In the case of gas chromatography, for analyzing fatty acids, but also in case of the risk assessment with pesticides, for example. And then for the third step, we have the bioactivities and the cell culture, and we develop, for now at least, only in vitro assays. For the in vivo assays, usually we perform collaborations with other institutes to test the anti-proliferative activity in tumor and non-tumor cells, anti-inflammatory activity, antioxidant assays that are the highest background in the case of Professor Miguel, and assays regarding real-time dose response, morphology, and cell cycle kinetics, using a specific technology to assess these capabilities and effects in the cells. So these are more or less the equipments we have for the microwave, pressurized liquid extraction, ultrasound, the high-pressure equipment that we have in our university. Then for the identification, the HPLC, the different HPLC, we have the MS, that is the equipment we most use for the polyphenols, and the gas chromatography coupled to MS for the fatty acids, terpenes, essential oils, pesticides. And then for the cell culture or the bioactivities in section 3, we have a specific room with level 2 security to develop the assays in antioxidant activity, anti-proliferative, anti-inflammatory. And here is the equipment I was telling you that allows us to determine the dose kinetic response, the cell morphology, and even tracking single cells during the treatment. And this is the video moving the cells. So as a resume, these are the three lines I was speaking: the extraction, identification, and bioactivities. And this is the complementary research line about optimization towards the industrial scale-up that we develop here with different kind of softwares. We usually use Excel, but also Design Spirit and MATLAB to achieve these optimal solutions for obtaining the highest extraction yield. So I think the easier now is to present some examples of our work, and I would like to first start with this, that was a scientific paper entitled Obtaining a Natural Additive Based on Catechins from Arbutus unedu, Competitive and Promising Extracts from Application in the Food Industry. Also, I would like to say that this article was transferred to a patent. This is a reference, and the idea of the work was to obtain these catechins from arbutus, from the fruits of the arbutus, by heat-assisted extraction and ultrasound-assisted extraction. We select the three most affecting independent variables in each process using a circumscribed central composite design and the RSM to optimize the process. So as a yield, we select the anthocyanin content that we were measured in different units regarding the fruit and regarding the residue, and the different catechins, as you may see here, delphinidine, cyanidine, and thyanidine, and the total of both compounds. So here you have the results for the extraction yield based on the dry weight of the sample after extraction, and in particular for both techniques in case of the fruit and in case of the residue. So for example, you may see here in this case in the fruit for the ultrasound assisted extraction that in the case of power and time it was favored when time and power were medium. However, you may see here, for example, that when regarding the time and the solvent, the samples always preferred the highest concentration of solvent. The global conditions that we find after the optimization for heating assisted extraction were the ones displayed at the slide. 20 minutes, 90 degrees, and 65% of ethanol in the solvent, different ones from the ultrasound assisted extraction, and the yields for each of the cyanidins and the total compounds. So in this case, we found that heat assisted extraction has the higher recovery rate, although we usually also study this from an industrial perspective regarding the consumption in terms of energy and in terms of solvent. As I was saying, this article is related to a patent publication. The name is more or less the same, obtaining natural additives with preservative function from Arbutus unedus. And then the idea of this extract was further to incorporate, that is also published in food chemistry, to some specific pastry product as a preservative for the antioxidant properties. And we could demonstrate that this natural extract was competing with the synthetic antioxidants to provide the same functions, but without the harmful effects that could provide the synthetic ones. This is another example, very similar, that we were working, in this case, not with catechins, but to obtain rosmarinic acid from Melissa officinalis. And we used three techniques: heat-assisted extraction, ultrasound, but we also incorporate microwave. This is the design for each of the variables that we use. For example, time tested from 30 to 150. Not the same in the case of ultrasounds because the matrix become destroyed, so the time has to be shorter. But then also for the temperature, or in this case, the power of the equipment. And in this case, the responses were the rosmarinic acid extraction yield, the purity of the compound, and the relation between these two concepts. So here were the results, the same as you can see in the pictures. Some of them were really good for the last response, that is at the end the one that we are most interested. And we could also study the patterns between the solid to liquid ratio in each extraction system to monitor and see how they affect the different extraction. In this case, as it was more or less seen from the pictures, the ultrasound assisted extraction showed the best optimization, and here with the best ratio between extraction yield and purity for rosmarinic acid. And in this case, it was the same. It was transferred to a patent publication consisting in the same preservative function for the antioxidant capacity of this extract. For the publications, we have many others you can consult in the internet, and I would like to present some projects because they are not so easily available in the website. So these are some of the ongoing research projects we have at this moment. The first one is a Europe Horizon wet biome, focusing on and revealing the potential of wet microbiome for the development of healthier, more sustainable and resilient wet-derived food and feed products. The second one is a national project focused on the fermentation of wine and beer bagasse to obtain novel ingredients and natural extracts. Then we have two projects in collaboration with the EFSA, the European Food and Safety Authority in Europe, one of them working with algae and the other with byproducts, but both of them regarding the risk assessment, and a project with Portugal for the hypocholesterolemic formulation from mushroom byproducts. So I would like to present one of them, in this case the second, that is the bagasse fermentation. And the objective of the project was to evaluate microorganisms, bagasse and fermented wine and beer residues or byproducts to develop new ingredients in the food industry and incorporate into food and beverage. In particular, the idea is to start with the bagasse, apply a fungus called monascus, that is the one obtained from the red rice from Asia. And while this fungus is growing and developing the fermentation, they produce a red pigment that can be applied in other products. And the idea is to develop novel beers, red beers that have this pigment, and also novel bread, especially for the ones that are rich in cereals and that had this color more red, but also novel food additives, because we are aware that they will have some antioxidant and antimicrobial properties. Yes, this project is now under development between the University of Vigo and the National Council of Research in Spain, in Valencia. And the idea is, as you can see in this picture, joining the upstream with the downstream, together with a focus on promoting the circular economy, stop composting the byproducts and use it for another purpose, and designing these new additives, but also new projects for bread and beer. And these are other completed research projects that we had in the past. Some of them, for example, related to prediction models for wines, characterization for extra virgin olive oil, a research network in the field of polyphenols, strengthening aquaculture in Latin America, an education project on the organic production of chestnuts, or an integrated approach for the sustainable food systems. We have more connected with companies, for example, with Algamar, that is a company focused on seaweeds, with Sea Lab working on fish and large fish because of the content in mercury. The App for Health was this big project, European, focused on the upcycling of olive, grape, and nut byproducts, and others. So for the scaling up, I would like to make a brief mention that we work close with the companies. We think it's a very important part of our work to transfer this knowledge. And at this moment, we are working with some of these. For example, this is Mercantia, working with byproducts from wine, from cocoa, from different matrices to develop functional beverage. With these two companies for developing system sensors to avoid spoilage in the fish, with Algamar focusing on obtaining extracts from seaweeds to apply it as functional foods, and then others with, for example, Abril, which is focused on olive oil, and Kiwi, focused on kiwi fruit. So I would like to highlight for sure the work with Algamar. That is a company founded in 1996 that promotes and spreads the nutritional properties of the native seaweeds from Galicia. We have developed together with them a pilot plant that you can see here to obtain extracts, in this case rich in fucoxanthin, a carotenoid from brown algae. And we had a project called Neurofood, which aimed to develop new functional foods and nutritive supplements aimed at neuroconnective development. And we develop these products in the bottom with as food supplements, all of them rich in this compound fucoxanthin that we saw the good properties for neuroprotection it has. Again, these are some pictures from the pilot plant, and here you can see Miguel with some of them of the people from the company. These are some publications we had related to the work developed with the seaweeds. And as the end of the presentation, I would like again to encourage you to contact us for more information and be part of our team. Sometimes we have chances to apply for doctoral grants or postdoctoral scholarships from the regional government, the national government, or even some European or connected initiatives. These are this is one picture of the research group in a beautiful place that was selected. And thank you for your time. Once again, I would like to apologize for Professor Miguel that could not attend today, but it was impossible for him. This is a picture of our city. I think it's very beautiful, so we would like to welcome all of you, and please feel free to send us any question you may have or asking me now. I will be more than happy to answer. Thank you.
Thank you, Mary. Thanks for your insightful presentation and the things which your institute is doing. It's very enlightening to know that you are doing a lot of analysis in the field of the botanicals, and happy to know that you are working with the industries closely so that technologies can be commercialized. Used for the business and for the society in a more effective ways. When an institute is associated with the industry, it knows the tidbits of the intricacies of that particular technology, that particular ingredients. What are the finer points which is required to be taken care of while producing the products out of that technology, out of that product. So it's very nice to know that we couldn't hear Professor Vico, Professor Miguel, but it's that you filled the space well, and you also presented and your institutes nicely. And so what are the other things? So I also want to tell you one thing. We in India have a lot of botanicals. We also do a lot of R&D. We have a lot of pharma companies. Those are working in these nutraceuticals and herbal products. And fortunately, by the initiative of our Prime Minister, we have also incorporated in our regulation and one dedicated regulation for these natural food products, which is called the Ayurveda Ahar. In India, we call it Ayurveda. So we have a food system which is based on the Ayurveda. So we have a dedicated regulation where we have given the reference to the modern encyclopedias, modern books, modern technologies, as well as our ancient wisdom books. So we have close to 70 ancient wisdom books where we have documented the effects and the nutraceuticals and the botanicals and their importance and their various uses in India. And we have, of course, a dedicated and shortly we'll be having a lot of dedicated institutes for doing all these research. And I'm happy that Spain is already doing all these things, and we look forward for a collaborative work with your institutes. And what are the other things beside all these botanicals your institute is doing? Are you doing some pesticides or, tell us something, in nutshell, the salient feature of how the Spain do for the food safety. See, as we have in India.
Thank you very much for the collaboration initiative, because we will be happy to collaborate, and also for the question. In the case of botanicals in Europe, the situation is we are a little bit collapsed. We have so many different products that the European agency is publishing. They call it Compendium of Botanicals, to summarize the different plants or botanicals they have, the parts that we are studying, the parts that have problems, because sometimes from one plant the root is fine, but the leaves have some problems with specific compounds or the opposite.
Right.
So they are a little bit collapsed in this sense. And for us, for our research group, besides from the botanicals, I would like to highlight the same. The work with them, with the companies, is very important for us because usually they give us which are their problems. And if we know the problems, we can know where to work. For example, for the fish, we are working now, they have a strong problem with the mercury, the concentrations of mercury in the big fishes, the ones that are traveling through big distance in the ocean because of the bioaccumulation. And we are now developing a project trying to extract this mercury from the fish because the companies, the fishermen are so interested in trying to find a solution. Like our governments are pushing down the limits for mercury because they have.
Yes, yes. Mercury is a global issue. We in India, we also have very stringent regulations on this angle. We also have methyl mercury in place. I hope your country is also having the regulations for the methyl mercury, which is a potent pollutant for the fishing products. So mercury, of course, is a very challenging and very important contaminant. So for this marine food is concerned. Yes.
Yeah. Thank you. And then for the I don't know, for the pesticides, we are working also in them, as we work a lot with the byproducts and with vegetal byproducts for sure. For example, with peels from fruits, from oranges, from apples. We are trying also to assess the pesticides charge and the risk assessment of this potential harm. In terms of safety, these are the two research lines that we are focusing the most. From one side, the heavy metals, and from other side, the pesticides. Also because our background is more in chemistry, so we always pushes for the chemical contaminants, not for the biological, for example, hazards.
Right. So do you have some regulatory body which governs all these regulations, which controls the limit of the MRLs of the pesticides, MRL of this heavy metals and other contaminants in your country, or you follow the EU guidelines?
We have both. We have the European agency that sets the European Commission that sets the limits, and we also have a national agency. It's called AESAN.
Yes.
AESAN, and they are the ones making the recommendations. Not always the regulations. For the regulations is the government, but they also make some recommendations. For example, with the seaweeds, the algae. They published recently another report we are very interested, that is the content of iodine in seaweeds. So we are also following the recommendations and try to approach from that point of view.
So how these industries in your country is collaborating with the government to set up these standards, as you told in your presentation that lot of this private industries are also part of your study on this. This is a very interesting plant in India also. We use this for the anti-mosquito repellent qualities. So it is called the lemon balm kind, some lemon balm, which on which the plant which you are working. So this is a very interesting plant, I would say, with a lot of terpenoids, a lot of botanicals can be extracted. So not many other plants are also there. So it is also used for some used for the aromatic therapies kind. So in India we have a robust documentation, ancient wisdom books, which gives us good insight for using all these kind of herbs, and which are less potent, which are of course we can always concentrate them, and natural to our physiology. So see, the thing is that we should encourage the things which are natural to our physiology, which your institute is doing. Your institute is doing so many all these kind of activities. So far this pesticides are concerned. How many pesticides are regulated in your country? Can you give me some rough idea, like 50, 20 or 200 or like?
At this moment, the problem is, for example, for our regulations follow the European community. And I don't know, I can say that, for example, now for active substance, there are approved about 400 active substances.
So they are they are the additives, or they are the some substance which are
They are
not
substances,
contaminated.
like a specific substance that are then included into the pesticides. So pesticides as formulation are less. I would say,
Yes,
I
yes.
don't know, maybe about 100, but
Mhm.
most of them, they will be under assessment the next year. So this is one of the main constrictions we have now, that we are looking for new alternatives, because the expiry date, let's say, it will finish next year, or they will be reevaluated, and it's very probable that they don't get the reapproval.
Sorry to interrupt. We have to take a break for lunch and start the next session. I thank Dr. SSR Raghav sir and Dr. Peter Associate Ms. Mariam for joining us online from Spain on such a short notice. Thank you, sir. Thank you, ma'am. Now we'll be taking a break for lunch, and we'll be resuming the next session on foodpreneurs at 2 p.m. I request participants and the students don't leave, because we will be announcing the winners for the oral and poster presentation online and offline in the validity session. And I request to kindly adhere to the timings I have mentioned. We'll strictly start at 2 p.m. after lunch. Thank you.
Thank you, madam. Thanks for
Thank you, sir.
giving this
Thank you, sir.
opportunity of addressing this gathering and interacting with the students of your institute. And congratulations that you are, celebrating this event 8 years in a row. And I hope that this tradition will continue for so many years with more insightful ideas, and will definitely come out. The outcomes of all the deliberations will definitely will prove beneficial to the students as well as to the society. Thank you all.
Thank you.
Thank you.
Thank you, sir.